Hello everyone, and welcome to today's Drug Target Review webinar called Early De-risking in Vitro Safety Strategies for Pre-IND Success. My name is Sam Whitewick, Editorial Assistant at Drug Target Review, and I'll be moderating today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for coming today. And I'm happy to be joined by Dr. Emily DeVos to discover how in vitro and computational tools reduce the reliance on animal testing and support smarter, earlier decision-making in drug discovery. Following the presentation, we'll move on to a Q&A session. So please submit your questions at any time during the discussion via the questions panel on the right-hand side of your screen. And with that, I'll hand over to Dr. DeVos to begin the webinar. Over to you.
Thank you for the kind introduction. I'm happy to be here today to share insights from a collaborative initiative I led over the past year. Our goal has been to improve early de-risking in drug discovery through strategy in vitro safety profiling to support more successful IND submission. At first, let's see the global context of early de-risking in drug discovery, how the secondary pharmacology supports it, and the evolution of the screening framework. As many of you know, probably the journey from discovery to drug approval is long, expensive, and risky. It can take up 15 years and cost between $1 to $2 billion per approved drugs. The attrition rate remains around 90%, with safety concern being one of the leading causes of failure, whether in preclinical, clinical, or even post-marketing phases. Drug discovery during drug discovery, lead optimization is the most costing stage. When toxicity is discovered late, especially during clinical trial or post-launch, it can dramatically increase cost and delay. Pretty sure that many of you know also that. So how we can improve it? A great example comes from AstraZeneca 5R framework, which significantly reduced their safety-related attrition from the 2010 years. By focusing on the right safety, so you can see it on the right side of the slide, they emphasized understanding secondary pharmacology and target abilities. Their approach led to a drop in attrition from 82% to 52% in preclinical stages, and from 30% to just 8% in phase 2 clinical trial. You can see on the left side of the slide directly. This translates into substantial cost saving and more efficient development timelines that are not shown here, but you can have the details in the dedicated publication. This brings us to secondary pharmacology, what it is. We can also call it in vitro safety screening and any different expression, including safety, profiling, screening, etc. It measures the compound effect at molecular targets distinct from the intent therapeutic molecular one. It's fast, cost-effective, and often including IND or CTS application. Beyond regulatory expectation, it provides a real value in safety assessment because target activity are related to physiological mechanism and some of them to toxic mechanism. It can help to anticipate clinical and preclinical adverse outcome and optimize the de-risking plan. There are no official lists of critical targets to screen yet, but hopefully publication exists and share screening strategy from pharmaceutical industry. One of the foundational framework was published by Bose and colleagues in 2012. It introduced a consensual list of 44 targets associated with adverse events and covering diverse organ systems with a major focus on heart and brain. This collaborative effort among major pharma companies helped standardize early safety screening and reduce adverse effects in marketed drugs since the target list has been widely adopted. More recently, Brennan and colleagues introduced a new framework under the DrugSafe IQ consortium. It added 33 novel safety targets comparing to the original framework. mainly kinases, but also with more nuclear hormone receptor or proteases, and then diversifying the target classes proportion. It also expanded organ system coverage compared to the original previous framework. They also introduced a two-stage screening strategy we will explain in the next slide and provide clearer guidance for regulatory submission and enhances early safety assessment. While industry impact remains to be specified, but this new information will certainly influence the future of early safety assessment practices by creation of specialized target panel. We can also anticipate a similar impact for the original framework with a better and early anticipation of clinical toxicities. So in practice, how the latest industry recommendations have reshaped the potential screening strategies. We will focus on the left side. One possibility is to use an early screening panel of 18 targets. They were selected based on their strong association with clinical adverse events and high hit rates in the IQ-JUSAFE database. This panel includes, as I mentioned before, GPCR and channel and so on, allowing rapid profiling of compound during hit to lead and lead optimization phases. These phases commonly contain several iterations. The second stage will use a comprehensive panel of 59 targets for later stage profiling before first in vivo studies with key lead candidates as example. Using this type of approach minimize the exposure of animals to compound, which is in line with the principle of the 3 R's in animal experiments. Another potential use is to build an integrated risk assessment with safety margin calculation against targets of concern to select the safest preclinical candidates. This panel continues to focus on targets with a link to potential clinical side effects as the 18 first targets. Other possibilities, so here we are focused on the right side of the slide. Other possibilities can be using the full panel of 77 targets to screen at one concentration during iteration cycles to select and optimize lead candidates and follow up with dose response curve for any considered drug candidates. To be noted that there is no single ideal strategy appropriate in all scenarios. Numerous approach existing. Some companies may have targets or have preference for binding on functional mode of assays. I will not detail here the consideration of binding on functional assays, even if it's an interesting question. This is not the topic of today. At Eurofins Discovery, we have integrated the original and new framework insight into our safety screen panel offer, enabling a flexible and scalable profiling strategy to adapt all strategies. We propose early stage solution, including the safety screen 18 core panel to engage earlier and fast safety profiling, or the safety screen 44 ideal for hit to lead phases and widely adopted in regulatory submission. In the mid-stage safety solution, we have the safety screen 59 and the 77. So as a reminder, including the latest recommended targets, which can support early hazard identification or support lead optimization. And finally, we propose broader panels to support more comprehensive profiling if needed. The latest created panels written in orange are proposing almost an all format again to offer flexible offer. Finally, let's touch on the evolving regulatory and ethical landscape in safety assessments. Historically, animal testing has been mandatory in pharmaceutical industry and pharmaceutical safety assessments. But recent change like FDA Modernization Act or the latest European Commission amendment allow now drug registration without absolute reliance on animals models. This shift is driven by growing pressure from public opinion and policymakers across industries, including cosmetics, chemical, and now extending to pharma. In this context, new approach methodologies, commonly called NAM, are gaining momentum. This includes in silico models, using AI, for example, in vitro system, mimicking human physiology, and chemical assays exploring molecular interaction as our secondary pharmacology panels, for example. So the desirable feature would be the replacement of animal testing, but one reachable step is to reduce and refine their use by implementation of alternative earlier in the drug discovery, and also improving the interpretation and prediction of these alternative methods. And to finish on this slide, the alternative methods has also an enormous cost-saving potential. As we move toward a broader adoption of in vitro safety profiling, one of the key challenges we face now is how to interpret the results to efficiently impact the drug discovery. Understanding target abilities needs a high amount of information, and in vitro safety profiling generates a large amount of data, especially when you're screening dozens of targets with dozens of compounds. It quickly turns into a relay lake. This is why we developed a solution we call in vitro safety insights. It displays pharmacological data from safety screen panels and help in interpretation, highlighting organ at risk and suggesting potential follow-up assays according to the identified hazards. I have to claim now that this tool should not be considered as a final guidance, but it offers a first step to support interpretation. And all this data should need contextualization to refine the mitigation plan. So how does it look like? You can see here the layout of the web interface displaying the result with interactive element designed to support interpretation, with as the top specification on the tool use the data it displays and the features, its map and a radar plot to display compound related result and to compare it, interpretation figure on the bottom, and on the right side you will have follow-up suggestion. Focusing on interactive results, you will find on the left side of the slide, you will find the inhibition percentage of control compound for each target, displaying a hit profile for each test compound. The red line at 50% level corresponds to the threshold where we consider targets as a hit. And on the web page, the filters are available to display what you need, so you can select one compound or several compounds depending on what you want to see. And target symbol represents the hits. The hit profile corresponds to an organ system hazard metric profile on the right side, which highlight the potential risk of toxicity in organs according to 3 levels of metrics: low, medium, and high. We explain later what does it mean. How the hazard metric score is calculated. It combines information from pharmacological results and data on association between targets and clinical and preclinical adverse effects. Targets adverse event association were obtained from 3 different sources. So consider safety pharmacology reviewed as Brennan 1, for example. Statistical targets adverse drug reaction association from proprietary and public database. And we perform a semi-automatically abstract review in PubMed on public resources. A corresponding level between 0 and 3 was defined, 0 to 3, I should say, was defined to describe the amount of evidence of adverse events in each organ system we have found. And for each target organ pair, the worst case scenario has been considered, taking the higher hazard matrix we calculated. So for example, if you, for one sources, you have found only one mention and one other you will have found more, we will take the worst case scenario. The score is then mitigated by a coefficient from target inhibition score, which were obtained with the attribution of a score to each range of target activity inhibition, from 0 to for a percentage inhibition lower than 50%, and to 1 for a percentage of inhibition higher than 80. For a compound, the highest organ system hazard metric score across all targets is retained to present the overall hazard level for each organ system, which is represented on the final figure on the right side of the slide. To complete our offer, we just have launched a new a dedicated visualization tool to interpret the safety scan 47 panel results, which includes the 44 consensual targets from the original screening framework and 3 additional kinases represented in the new screening framework. So it offers 3 key features. You will have radar plot to visualize compound activity across the full panel, safety margin analysis to highlight targets at risk and on potency and expected exposure, and at last a custom curve fitting tool for potency calculation. This tool, as I mentioned, complements our broader visualization strategy and helps in contextualization of pharmacological results. To illustrate how this strategy works in practice, let's look at case studies using the in vitro safety insight tool, applying the new screening framework to a set of antidepressant compounds. So we conduct a case studies using 8 marketed antidepressants from different classes, tricyclics, second-generation compound, and selective serotonin reuptake inhibitors group. So I will call it during the presentation SSRI. These compounds were screened using safety screen 18 and safety screen 59 panel at 10 micromolar, following the IQ safe recommendation screening mode. So the goal was to compare the obtained hit profile and organ system at risk using the in vitro safety insight tool to those found in literature. On the slide, you will see the radar plot at the top for each categories. So first on the left, the tricyclic, on the middle, the second generation group, and on the right, the SSRI group. At the bottom, you will see in the same order the organ system hazard metric profile, which has been slightly shifted between compounds to better visualize overlapping data points resulting from similar pharmacological profile, because you can see briefly that a lot of them have a similar hit profile and a similar organ system hazard metric profile, especially the tricyclic agents, so amitriptyline and imipramine, and the maprotilene and amoxapine from second generation compound. These compounds are known to share common side effects such as related to cardiovascular system, sedation, or motor dysfunction. Among the second generation compound, bupropion is an uncommon antidepressant known for high risk of seizure like the maprotilene, but with minimal cardiovascular effect. And this is something we can see clearly on the organ system figure, hazard metric, organ system hazard metric figure, where we can see that the bupropion is really different compared to the other in the same group. Regarding the SSRIs compound, they show different hit profile compared to the other antidepressant class. They are known to induce minimal anticholinergic effects and less sedation then, as a consequences. The best known side effects of this class are gastrointestinal upset, sleep disturbance, weight loss, and so on. I will not detail all because the list is quite long. In the SSRI group, fluoxetine show more off targets in this safety panel with cardiac-related effect, which is quite consistent with what we are saying on the figure, and correspond to his in literature to his non-risk in a torsade de pointe occurrence, which is higher for citalopram and fluoxetine. Then, using the safety screen 59 panel to complete the global analysis of each class of antidepressant compound, because as you may have understood now, the safety screen 80 will not be enough. You need to complete the analysis. So we use the safety screen 59. We screen all these all the compound. And again, we can see high amount of similarities in heat profile for tricyclic and maproptyline and amoxapine compounds in the second generation group. We can also note that bupropion stays atypical with respect to intended activities at targets, as example, no serotonin reuptake transporter. And we can also see that the SSRI profile stay heterogeneous between them, showing expected activities at targets as hopefully the serotonin reuptake transporter, and only at the 5-HT2A for fluvoxamine. Whereas fluoxetine and citalopram display additional hits on muscarinic receptor and others that I will not detail because it will be too long and not really useful for this presentation. Anyway, what are the results and the corresponding organ system as a metric figure after this heat profile? So here you have the corresponding figure for each classes. At first view, as seen for the safety screen 18, we keep same profile for tricyclic agent maproptyline and amoxapine from second generation group due to their similar heat profile, no surprise. Additional information compared to the previous panels. So I think it's better to display the figure to remind you, or it will be easier to compare. So I was saying that additional information compared to the safety screen 18 were when we are comparing between panels results. We can see additional identified hazard for bupropion in GI tract as example. It is consistent with the additional hits we have seen in the heat profile on kappa opioid receptor, which is associated with gastrointestinal hypomotility as example. Regarding SSRI class, additional activities made appear hazard in embryonic development organ system. It is known that the use of SSRI during pregnancy affects the development with premature birth or decreased body weight on the child or growth retardation. So again, we are consistent with the expected activities in literature. So the conclusion of the case studies, as we have seen it, that we can say that pharmacological results and heat profile were consistent with previously reported data in literature. We have seen that the organ system as our metric effectively distinguish between different classes of antidepressants, highlighting organ system that may be at risk according to their chemical structure. I put in orange the chemical structure on the right side of the slide. So to be very honest, I'm not a chemist, but just mentioning, I've seen that in the literature, they were mentioning how they improve the tricyclic to the second generation compound chemically, and it's feasible when we are exploring the heat profile and then the organ as our metric profile. Finally, they are close, but what we have seen is representative of optimization, which has been done before between first and second generation of antidepressants. This is something I forgot to mention just before, but we can improve the scoring prediction performance because sometimes we have seen some little discrepancy between the level, so the number of probability of occurrence of adverse events, which was not really correlated with the level of hazard metrics. For example, we have seen for fluvoxamine, which should present highest level of hazard metrics in GI organ system. It was not exactly like that because the other SSRIs display higher hazard metrics than fluvoxamine. So this is something we can improve to have a better scoring prediction. but it also gives information on how to use this tool and what are the limitations. One thing we can improve in the future to have more prediction performance is to refine the classification of adverse events in organ system. We can also anticipate to include additional quantitative parameter as well, potencies and safety margin, for example. We can add more targets in secondary pharmacology routine testing, or include more contextual information, but this is something which will be very difficult in a tool. But we have to consider this. To conclude on this webinar, our tool facilitates a quick overview of the secondary pharmacology results and the corresponding organ at risk profile, aiding in the ranking and prioritization of compounds. When combined with time-effective target profiling, it can significantly improve early drug discovery stages. It also facilitated hazard identification, especially with hazard matrix level in organ system. And again, I mentioned and insist on the fact that it's only and mostly identify hazard risk. And for an integrated risk assessment, more quantitative information with potency, and again, I repeat, anticipated exposure to calculate safety margin at targets could be very useful. This will consist in our next improvement of the tool in vitro safety insights. More target-related information as the tissue distribution or target homology or across species can help to refine the risk mitigation. And at last, we can also say that contextualization is a project environment, as disease population can impact the risk-benefits balance evaluation. As we look ahead, the integration of strategic in vitro safety profiling and smart visualization tool offer a powerful opportunity to anticipate risk earlier in the drug development process. This approach not only supports ethical innovation, but by reducing reliance on animal testing, but also accelerates the path toward safer, more cost-effective medicines. To further improve this strategy, we will need to develop specialized panels for specific toxicity mechanisms such as seizure liability. We can also continuously monitor emerging data on target-related adverse events to complete our safety assessment. And we can also obviously improve our computational tool to improve prediction accuracy and support decision-making. Together, this improvement will help shape a future where safety assessment will be more predictive, more ethical, and more efficient. As takeaway of this webinar, I hope I convinced you that now that drug attrition remains high. Okay, everyone knows it, but no hits. And I hope I convince you that early safety profiling is essential to save costs and to be more ethical and so on. Of target screening frameworks with the original and especially with the new, with the safety screen 18, 59 and 77 panels is cost effective and can really improve compound prioritization and help to anticipate clinical outcomes. Our visualization tools can really support decision making and risk assessments. Regulatory shift towards a new approach methodology also opened new opportunities for non-animal safety evaluations or more ethical research. And in the future, we can anticipate to refine scoring, expand targets, we screen and integrate more contextual data for better risk prediction. Thank you for your attention, and now it's time to question. I will be happy to answer.
Thank you very much, Dr. DeVos, for that fantastic discussion. Yeah, as the slide says, we'll now open it up to the Q&A session. So a quick reminder that you can still submit your questions using the questions panel on the right-hand side of your menu. So kick it off with the first question, which is, what exactly does the screen consist of? binding. Eliza, if a compound has a high score in the radar plot for a target, what does it mean?
Everyone, so thank you for your question. So yeah, for here we present data from binding and enzymatic assays. And your second part of the question is a compound with a high score. Okay, in the radar plot, so you mean regarding the pharmacological data. So in the radar plot, we are presenting the percentage of inhibition of the control compounds. Well, tell me if I'm not clear enough.
Yeah, I think that's perfect.
Yes.
Yeah, no worries. I'll move on to the next question, which is what are the key considerations when choosing between the different safety screen panels for early in vitro safety profiling?
So okay, for early in vitro safety profiling, so here the choice will depend on the stage of the drug discovery process. So early means it could be at different stage depending on the company. But for early, we can also consider the chemical serum maturity and resources, obviously maturity availability. So in this presentation, we shared a new framework strategy. So if you want to begin very early in the drug discovery, you have a very affordable with short TAT panel with the safety screen 18. You have the more critical target with a high hit rate in this panel. So this is something you can consider if you have a short budget at beginning, but just keep in mind that it will not be enough at the end of the in vitro screening strategy. So you will have to complete with the safety screen 59 if you are choosing safety 18 first. You can also take the original framework, the safety screen 44, safety scan 47 could be good also. And you can also choose to follow the new recommendation, which would be adopted in the next years if it happens as for the first framework targets. After, the other consideration will be, should we use the binding mode? Should we use the functional modes at first with the dose response curve or one concentration screening? So as I mentioned in one of my slides, there is no one good strategy again. And sorry for confused answer, it's mostly case by case at the end. So if you need some guidance, obviously there are several specialists to do it, and we can offer this if you have more specialized case question for your project. We can help you to choose among our offer.
Great. The next question here is, what are the main technical challenges in interpreting off-target activity data, and how does the in vitro safety insight tool help streamline this analysis for project teams?
So more technical, yeah, the main technical challenge when you try to interpret the target-related information is a huge amount of target-related information, and to choose which one is relevant for your interpretation. So you will have several challenges. You will have data overload. So for human brain, could be very nightmare at the end. If you have only one target to address, okay, could be easy. But if you are considering 1, 2, 3, up to 10, and several compounds to compare, it could be very difficult. Other things that could be tricky is the lack of context regarding the target relevance. Sometimes you can find the activity on the targets. We anticipate this activity related to a potential clinical outcome. The question would be, okay, will it happen really? So you will have to go further with more information. So regarding the probability of occurrence of the clinical outcomes, and is it really serious for your project? So far, what we are addressing with the in vitro safety insight tool is mostly to save time in information to have information related to the targets, and we offer an opportunity to have a summary of what could happen in organs. Obviously, you will need more information regarding targets, and this is something that we can add in the next improvement of the tool. Yes, and yeah, I think all. Yeah, that's all.
No worries. Thank you. And this next question sort of has quite a few parts. So for the in vitro safety insight tool, how could we have access to related adverse events in organ systems?
Okay, so if we are talking about how can we have access to the data we have, this is something we are not offering yet. So currently we have the data. So to create the system of organs and so on, obviously we had to classify the adverse events in the different organ systems. And so we have the database, but we have to create an interface which is really human friendly, not only for a computer. And so yeah, so far we cannot offer it, but yeah, I hope it will come very fast.
And then do you provide the target name related to each organ risk in the tool?
I'm sorry, can you repeat?
Yes, the next part was, do you provide the target name related to each organ risks in the tool?
Oh yeah. The simple answer is no. We can also, we have the information, but so far the tool is not displaying it.
And then how do the tools determine which potential follow-up assays to perform?
So yes, I did not detail it in the presentation. So we present some suggestion of follow-up assays regarding your results after performing a safety panel. We group the potential follow-up assays according to each system of organs. So across all the reference discovery offer. So it means that depending on, for example, if you have some findings in cardiovascular organ, we group all the assays related to this organ system. So it means that it's just suggestion, and it means that you will not have to do all of this. It's just to give an information that we are able to support if you want to follow up on it.
Great. And the next part, how were the organ systems created in the in vitro safety insight tool to classify adverse events?
The organ system. Okay, so we try to follow the logical of the major ACLS classification. We did not choose exactly that, but the aim was to cover the full body of a human. And so yeah, we organized the each system across. Yeah, it's not very clear what I'm saying. It's just using, yeah, we just group in different organ systems covering the full body. And so yeah, we were inspired by the major classification, and we just exclude which was not applicable to the tool and to the secondary pharmacology interpretation. So for as example, it was investigational, for example, is a group in major classification which is not relevant for secondary pharmacology.
Thank you. And the next part, were there any specific organ systems or adverse event categories that were particularly challenging to classify or group in the system of organs?
Yes, it's related to the previous. Yeah, something I forgot to mention just before, when we created the organ systems, we after to sort the adverse events, you have to determine how you will classify it. So we classified regarding the location of the outcome more than the etiology, but sometimes it was not feasible to use other things than etiology, for example, for cancer, oncology, or genotox. And other category which was difficult was regarding the metabolism outcome. It could be very broad, could be related to the hypophysis, which is in the CNS, for example, or related to surrenal glands, which are completely in other parts. So yeah, regarding the classification of this adverse event was really, yeah, hard to do, and this is something I think we will improve with the next version. I think we will create a special organ system for metabolism adverse event.
Great. And the next question here, what improvements do you see as most critical for increasing the predictive power of in vitro assays and computational tools in candidate drug selection?
That's lots of improvements. We have long roads to do it. I think we are not the only one person working on it. One thing I see first is using more say quantitative parameter. So far, we created a semi-quantitative parameter using the score adapted to percentage of inhibition, but more efficient prediction. The prediction power could be more efficient if we are using potency and safety margin analysis, since if you have ideas of safety margin analysis at the targets, you can translate, and I say it with caution, you can translate on a kind of safety margin on adverse outcome. The main issue with this type of reasoning is that we are not sure for all this association between adverse outcome and targets, which activities is really related to which occurrence of adverse events, for example. So, and currently there are no official response anywhere. So everyone is working, I think everyone in the domain is working on it. So we have a huge work around this. I mentioned it during my presentation. It's not only about how much the target is related to the adverse outcome, it's also the distribution of the targets. For example, if you are using a compound which distribute very differently than another, you will not have the same activity regarding the target distributions. Obviously, you will have a different risk after. And yeah, there is so much things. And again, yeah, one thing interesting to consider also is that we are working on only what we know, meaning that we are testing targets we know that they are related, so the others we ignore, and we ignore sometimes physiology and we ignore also pathophysiological pathways. So if we ignore this, we cannot know which target to address and so on. So obviously we need to continue to study the adverse outcome pathway. I think it's not completely, it's very, very long discussion. This my first thought.
Yeah. The next question is, have the visualization tools been tested or validated using external client data or real world case studies?
Yeah, so we have tested, so the visualization tool have been launched at the beginning of this year. So we have tested on the with the case study, and our client began to use it. So yeah, since the beginning of the year. So far we don't have much more feedback on how they used in their project context. We are waiting for some feedback. So far they are good, but we don't have so many details on how it works with their context, their project context. But we'll be happy to discuss about this later, and we will work in more case studies also, obviously.
Great. And the next question, based on the free drug principle, only the unbound drug can bind to the target and off targets. Does it apply to this case? Do we need to correct do we need to correct for the free drug fraction across the tissues organs?
Okay, so I will take time to.
No worries, no problem.
Yeah, based on the free drug. Yeah. Yeah. In this case, my issue with this question is that in this case, which case are we talking about? So if Jana can just specify.
If you could just specify.
Are you talking about the score in this tool, or are you talking about another thing when I was discussing about parameters we have to use? I don't know if you can read it.
Just give you a couple of seconds to reply if not, but because we are running out of time. So I think if we don't get it there, I'll just wrap it up because I'm just conscious of the time. So yeah, unfortunately, that is all we have time for today. I just want to say thank you to Dr. DeVos for a fantastic discussion, and thank you for everyone for joining us online and for submitting your questions as well. As you leave the webinar, a short survey will appear on your screen asking you to rate your experience. If you're unable to complete it now, it will be sent to you by email shortly, and we would greatly appreciate any feedback. The webinar will also be available on demand too. So on behalf of Drug Target Review and Eurofins Discovery, thank you for attending. And also apologies just before I do that, but I think there has been she has replied. Do you want to quickly answer that before I sign off, or shall I just end it there? So I'm just conscious of the time.
Okay.
We have a bit of time, so sorry, Dr. DeVos, if you want to just answer that question for Jana.
Okay, so yeah, you were okay, we'll take it. The translation of the in vitro data into clinical risk. Oh yeah. And the first question was based on free drug, yeah. Do we need to correct free drug? Yeah. my simple question would be yes, you're right. So far when we are talking about safety margin, obviously it's better to take the free the unbound drug. So when we are talking about the translation from in vitro to in vivo, obviously it's better to take the free part of the drug. I hope it will. But again, if it's not satisfying, or if there is a misunderstood, because I'm saying I'm reading the third part, do not hesitate to contact me. I will be happy to discuss with you if it's not enough.
That's great, thank you. So just on behalf of Drug Target Review and Eurofins Discovery, that is the end of the webinar. Thank you for attending, everyone, and we hope to see you all again soon. Goodbye.